Our project focuses on the application of self-supervised multimodal learning. We are undergraduates from the Department of Electronic and Telecommunication Engineering at the University of Marachua. Our project is motivated by the difficulty of labeling complex data such as 3D point clouds and high-dimensional spatial gene expression. Therefore, transitioning to the paradigm of self-supervised learning is important, which does not require explicit labels. Our general problem statement addresses how to effectively segment unlabeled multimodal data. In life sciences, integrating into histopathology and spatial transcriptomics, and in physical sciences, combining rendered images and 3D point clouds. Our objective is to develop effective end-to-end self-supervised models for both these areas, addressing the current limitations. Spatial transcriptomics model architecture. Spatial transcriptomics provide complex, high-dimensional data on gene expression distribution, which is challenging to annotate but highly valuable, particularly in cancer research. Analyzing this data is crucial for understanding significant challenges like tumor heterogeneity, which contributes to treatment resistance and immune evasion, and the role of the tumor microenvironment, both of which are essential for effective cancer analysis and treatment strategies. We have developed a novel architecture that efficiently learns a joint representation from spatial transcriptomics and histopathology images. A graph structure is created using the spatial locations of the spots. Expressed genes are added as feature vectors for each spot. This graph is then processed through a graph transformer to generate RNA embeddings. Simultaneously, the histopathology image is divided into patches and passed through an image encoder to create image embeddings. These RNA and image embeddings are then integrated and processed by a shared encoder to extract a joint latent embedding for each spot. This shared embedding is then clustered and visualized in a dimensionality-reduced way. The resulting cluster assignment for embedding determines the segmentation class in the spatial domain. The spot embeddings are mapped to the spatial domain and registered onto the histopathology image to generate the final output. We evaluated our model on a standard study cohort of breast cancer patients commonly used for benchmarking. Quantitatively, our method achieves performance that significantly surpasses current state-of-the-art models. Quantitative results provide a fundamental understanding of superior performance. As shown in the figure, the pink cluster in the ground truth represents invasive cancer at the core, and the red cluster represents cancer in situ, exposing the edge. Our model uniquely identified these two critical regions associated with tumor heterogeneity. While our self-supervised model identifies spatial regions within the tissue, these clusters don't come with pre-assigned biological labels. To understand what these regions represent, we perform a post-clustering analysis. We use the Wilcoxon test, a statistical method to find marker genes uniquely expressed in each cluster. By querying external gene databases, we uncover the biological identity and functional significance of these identified regions. Our second application is point cloud model architecture. A point cloud is a collection of 3D data points representing the external surface of an object or scene. Our RNDPMAE is a multi-branch masked auto-encoding framework integrating a point cloud encoder with 2D pre-trained RGB and depth image encoders. The point encoder utilizes a 3-stage hierarchical transformer processing 2048 input points. Its RGB and depth branches employ frozen pre-trained backbones, with 75% patch masking applied to RGB images. During pre-training, RNDPMAE reconstructs masked point cloud portions while applying 2D semantic-guided masking from depth data and multiscale contrastive learning between embeddings from visible point tokens and RGB image patches to enhance 3D representation learning. For downstream tasks, only the pre-trained point cloud encoder is retained for fine-tuning. RNDPMAE demonstrated strong performance across various evaluations, including linear SVM and full fine-tuning for object classification, achieving high accuracies on ModelNet 40 and ScanObjectNN and consistently outperforming prior methods. It also showed strong capabilities in part segmentation, indicating its effectiveness in capturing both high-level semantics and fine-grained geometric details. Our project successfully addressed challenging data analysis problems in spatial transcriptomics with the SENCA-st model and 3D point clouds with the RNDPMAE model. Both models outperformed existing methods, validating the effectiveness of our self-supervised learning approach for extracting insights from complex, unlabeled data.